And then now you can hit it, okay? Great.
Yeah. Okay, so as I said, I'm going to be presenting a little bit about cyclin-dependent kinase 12, which is also referred to as CDK12. Here is the AlphaFold model of my kinase of choice. CDK12 is commonly known for the role in phosphorylation of the C-terminal domain of the RNA polymerase II, which then upregulates the expression of genes involved in DNA repair. Depletion of CDK12 in cells does not alter the universal transcription rate, but at low doses of inhibitors, it can downregulate the expression of DNA repair genes. And at higher doses, it can also affect the expression of super-enhancer-associated genes, as we will see later on in my presentation. It is indirectly involved in the alternative splicing, specifically by processing splicing factors such as RSSF1, which are crucial in the processing of other proteins. CDK12 is a member of the CMGC kinase family, and as the name implies, it is a cyclin-dependent kinase. We can see from the phobias results down here that with a line at 1 for the non-cytoplasmic regions, it is a non-cytoplasmic protein, and all the other prediction lines are at 0, which indicates that it has no transmembrane or signal peptide regions. It is present in the nucleus of all tissues. It is universally expressed, but the higher levels are seen in the testes, ovaries, adrenal glands, and in cancer cells. This kinase is a fairly long protein. It has 1,040 residues, and the kinase domain spans from residue 727 to 1,020. This is the only order region of the protein, as we can see from the AIU prediction of intrinsically unstructured regions. As we saw earlier, the kinase has no membrane-spanning regions or lipid modifications, but as reported by Phosphosite, it counts with a wide range of post-translational modifications, including acetylation and glycosylation. Phosphorylation is definitely the most common PTM, and it is seen throughout the entirety protein sequence. Because of its involvement in DNA repair, alterations of CDK12 are commonly found in different malignancies, such as ovarian and breast cancer. In ovarian cancer, loss of function mutations in CDK12 is seen in 3% of the cases, and it is one of 8 genes that are recurrently mutated in the disease. Additionally, 50% of patients exhibit deficiencies in the homologous repair system, responsible for repairing DNA after double-stranded breaks. This suggests a strong link between CDK12-induced expression of DNA repair genes and ovarian cancer. In breast cancer, for ER-positive tumors, which have excessive signaling of estrogen receptors, these are typically treated with receptors inhibitors such as tamoxifen. However, downregulation of CDK12 has been reported to activate other self-promoting pathways, leading to the loss of dependence in estrogen for tissue growth. In these cases, sadly, the tumor becomes resistant to tamoxifen treatments. For HER2-positive cancers, on the other hand, we actually see the opposite. We see an overexpression of the human epidermal growth factor receptor 2, which is a super-enhancer-associated gene. This is likely caused in part by the increased levels of CDK12 reported in 71% of the cases. New treatment options are currently being investigated for all of these 3 diseases. For example, the inhibition of PARP1 alternative repair systems of the DNA damages, which would act after CDK loss of function. For ER-positive breast cancer, they are investigating the usage of dual treatment strategies that target ER signaling and the newly activated growth pathways. And finally, for HER2-positive breast cancer, they're looking into CDK12 inhibitors that can downregulate both the expression of HER2 receptors and the CDK12. And with this, here are my references, and thank you very much.
Okay, great talk. You did a great job. I want to open the floor for any questions. Remember, you are being graded on participation. There's a couple of questions there in the chat space, Alicia. Can you see them?
Not right now, but let me...
Okay,
maybe
Tiffany
if I stop
asks,
sharing.
Tiffany asks, can you explain more on how the different treatment options would work?
Oh yeah, definitely. I'll actually go back. Maybe I'll go back to the last slide. So basically what happens is for the ovarian cancer, because we have loss of function of CDK12, other DNA repair systems are going to kick in. But the idea is that if you have CDK12 loss of function, and then you also treat the cells with inhibitors for those alternative pathways, then hopefully the cell would just eventually die because it would accumulate too much DNA damage. For the ER positive ones, it's pretty self-explanatory. We have to target not only the original estrogen signaling that was, again, initially promoting the growth, but we also have to target the second ones that are now coming into play, like MAPK pathways. And finally, because in HER2 positive cancers, we have upregulation of CDK12, which is
Okay,
causing.
that's great. You've done a great job. I'm going to keep moving because you've done
Okay,
a good job, and I want to give Alison the floor next. And please do answer the question from Pearl in the chat space as well, Alicia.
sounds good. Thank you.
Alison, can you share your screen? Thank you.
Yep. Okay. You can see that?
Perfect.
Okay, perfect. Okay, the kinase I've chosen to study is CLK3. This is a CDC2-like dual specificity kinase that acts as both a serine threonine kinase as well as a tyrosine kinase. CLK3 is expressed in a wide range of tissues, as represented by this top figure here. It is typically localized to the nucleoplasm of the cell, but it is also localized to intermediate filaments. The function of CLK3 is to phosphorylate SR proteins, which are involved in the control of RNA splicing. The phosphorylation of SR proteins changes their distribution in the spliceosomal complex, which regulates the alternative splicing of pre-mRNA. This redistribution represents a difference in the number of splicing factors in the nucleoplasm, which could influence splice site selection of pre-mRNA. CLK3 includes a protein kinase domain, as well as a low complexity domain at the N-terminus end of the protein. 2 ATP binding sites and an active site at aspartate 283 have also been characterized for CLK3. There are many confirmed structures for CLK3 in the protein database, including the crystal structure shown here. What differentiates the CLK protein family from other kinases acting on SR proteins are features in the C-terminal domain, such as unique motifs and insertions. Although all CLK proteins have very high structural similarity. Through comparison of results from Phileas, Phoebus, Modo, Swiss Palm, and Deep Coral, I believe with high confidence that CLK3 does not have any transmembrane regions. Swiss Palm indicated a palmitoylation of a cysteine residue at position 4 of CLK3, but this could not be confirmed as there was no supporting literature. The results from Deep Coral indicate 2 possible coil-coil regions, but these are likely false positives as these are within the kinase domain. The plot obtained from Phoebus also shows that there is very low probability of there being any transmembrane regions. Additionally, the Modo results show that there are 2 very significant peaks in the plot, with the first peak being an arginine residue at position 12. However, this is likely a false positive as this is within the disordered domain. The other peak, however, consists of 2 residues, a tyrosine and a lysine at positions 272 and 273, which could be more significant as these are within the kinase domain. This could be a membrane interacting region, but not a transmembrane region, as there is not a significant amount of residues indicated to be membrane binding. There are no known PIP or MET stops characterized for CLK3, as it does not have any membrane binding regions. However, I investigated the Modo results to determine if the possible membrane interacting region is involved in regulation of CLK3. These residues are generally close to the active site and structure, but there is no evidence of CLK3 being phosphorylated at this position in literature. Through using disulipred, a possible lipid binding region was identified in the disorder domain, which correlates with identified phosphorylations of 2 serine residues at positions 76 and 78. There is a possible PIP stop here, but there's also minimal data to confirm this, as there is no supporting evidence of this being involved in regulation. Recently, CLK3 has been investigated as a potential oncogene in digestive system cancers such as cholangiocarcinoma or CCA. An overexpression of CLK3 has been observed in patients with CCA, which correlates with an increase in mortality rate. A missense mutation of glutamine at position 607 within the kinase domain has been identified that upregulates CLK3 and therefore indicates CLK3 as a direct oncogene. CLK3 is also overexpressed in other digestive system cancers such as colorectal and liver cancer, but the specific mutations are less completely characterized. And there are my references.
That's great, Alison. There's a question in the chat space from Caitlin. Is there a possibility that CLK3 could be investigated as an oncogene in other types of cancer?
I don't know for sure because all I could find was about digestive system cancers, but because CLK3 is expressed in so many tissues and it may, here, I'll go back to it. Because it is expressed in so many different types of tissues, I believe that it could be investigated as a potential oncogene in many different types of cancers.
Great. Could you answer the other questions in the chat space, please, Alison? And then I'm going to ask our next speaker, Ashwin, to share their screen. That was great. You guys are doing a great job. Ashwin, I think we can probably hear you. I can see your screen coming up.
Can you see it?
Yep, feel free to start.
Oops, that's the wrong one. Oh no, you can see it. Okay, so my presentation is on NEMA-related kinase 10 or NEC10. So the function I just drew out a little kind of process map that shows what's going on. I'll go over it. So this is the UniProt ID for any reference, and how this protein it responds to UV radiation. That's what activates its function. So when it's irradiated with UV, it complexes with MEK1, which is another kinase, and RAF1. And so MEK1 is mitogen activated protein kinase 1, and RAF1 is RAF proto-oncogene serine threonine protein kinase. NEC10 phosphorylates P53, and MEK1 phosphorylates ERK12. This is when they're in the ternary complex, like in the middle, after UV radiation. And so what both of these actions do is it arrests the cell in G2M phase. And this arrest is a pretty important step in the cell life cycle because it allows cells to repair DNA instead of dividing. So it's like a quality control, the cell checks if everything's okay before it divides. And this prevents cancer, like it prevents damaged DNA from replicating, which causes further issues. And some chemotherapies actually use this mechanism. So the expression, it's present everywhere in a lot of different tissues, but high expression is found in the testes and brain, and according to Protein Atlas, and according to different sources I found throughout like the research, it's also present in the lungs. It's localized to the nucleoplasm and the vesicles. And so according to Phoebus and Deep TMHMM, there is no evidence for transmembrane domains, which kind of makes sense because it's in the nucleoplasm. And so for the structure, this over here on the left is just the AlphaFold 3 structure. It's 1,172 amino acids long. This serine threonine kinase domain is from 519 to 712. And there are several low complexity domains throughout. And so also M magnesium 2 plus is a cofactor. There are no lipidations or no evidence of lipidations for these four fatty acids. There are many polar residues from 919 to 945. I didn't find a lot of research on this specific site, but it could be a binding site of some sort. And there is limited research on ligands, but we can assume that ATP is one of them because it's a kinase. But yeah, I didn't really find any papers on the actual ligand, people researching on it. So for the regulation, there are 3 sites within the kinase domain for phosphorylation, which is at these 3 locations. This is likely involved in the kinase reaction of the protein. There's also 2 phosphorylation sites outside the kinase domain, which is these 2. And according to a paper I read, NEC10 autophosphorylates to activate it, so these sites could be involved. And there's also a ubiquitination site at lysine 42, So this could be a signal for protein degradation, or I didn't find a lot too much about it, but usually ubiquitination signals protein degradation. So that's what I assume. And so for disease, there's really only one research disease, and it's ciliary dyskinesia, which is a disorder of the lungs. Basically, this means the cilia don't work properly, and this leads to chronic inflammation and bronchiectasis, which is, as you can see in the picture here, is just the airways getting widened and disfigured. This is an autosomal recessive disorder with two mutations, proline to leucine and arginine to cysteine. And this could be linked to cancer, not this disease, but the protein itself. Any malfunctions of the protein could be linked to cancer as well because of its involvement in the G2M arrest. And yeah, that's pretty much it.
Great, thanks, Ashwin. There's a question, a number of questions actually. I'll ask the first one for you. Do you know how the UV radiation activates the kinase?
Um, I tried to look for papers. I didn't really find the mechanism. It looks like the UV radiation causes the proteins to associate, like the three proteins, because that's how it kind of activates the ternary complex. But the papers from what I read, it just said it's kind of that's what happens. They didn't really find a mechanism, or it's not elucidated yet.
Okay, and are there any interactions that might help your kinase cross membranes
Um,
with other proteins that help signal its destination, for example?
I did find that it could be localized to vesicles, so there could be some transport between like the endoplasmic reticulum Golgi and that kind of sort to bring it out because since it's involved in a cilia disease of the lungs, then it could be affecting the extracellular space. So that could be how it gets there. But
Great.
yeah,
Okay, I think we're gonna have to move on just because you've done a great job and there's more questions that you'll need to answer in the chat space. Bryson, are you able to share your screen? Thanks. The talks have all been great. You guys have
Yes.
done a great job. Just share your screen and you'll be good to go.
Let's just get it up here. Sure. There it is.
Okay, great. You can start whenever you're ready.
Okay. For the diet kinase assignment, I chose to look at VRK2 or vaccine-related kinase 2, which is a serine threonine kinase involved in various signaling pathways. There are two major isoforms of VRK2, which correspond to their expression in various subcellular organelles. VRK2A is the predominant form, and it is expressed globally in all cell types. It attaches to the ER membrane and the mitochondrial membrane, and is most prominent in cells that have a high proliferation rate. This expression was also determined experimentally and confirmed with deep lock here. Phoebus also predicts that this is a non-cytoplasmic protein, as shown in the next slide. VRK2B is only expressed in small amounts in the cytoplasm and nucleus, and it lacks the transmembrane domain. The protein kinase domain in blue, which contains the active site, has been determined through crystallography. An AlphaFold can be used to show the transmembrane linker domain here in red. This transmembrane domain was determined using Phoebus and disoliprid. Disoliprid also shows that predicts residues that may interact with the membrane lipid surface. VRK2 is involved in many different signal transduction pathways shown here, but many more have also been identified. Phosphorylation of the barrier to autointegration factor at the N-terminus and P53 at threonine 18 are the predominant reactions in healthy cells. But ultimately, all of these pathways that VRK2 regulates are related to apoptosis or the inhibition of cell growth. The active site can also be seen here, with aspartic acid 166 being the specific active residue that acts as a proton acceptor when phosphorylating. VRK2 is regulated by RAN and GDP, like VRK1 shown here. It is assumed to block autophosphorylation activity of threonine residues, thus preventing its activation. It is associated with residues 256 and 320, as also shown in the previous diagram. VRK2 also exhibits minimal post-translational modifications as predicted by phosphosite and p-cismod, but it predicts a high level of phosphorylation and ubiquitination sites relative to the other modifications. p-cismod also predicts that only 4 cysteines can be modified with various modifications, but no other prediction software assumed there were any, so this might be a false positive. Phosphorylation most likely activates the kinase, and ubiquitination likely acts as a degradation signal, as the ubiquitin is added to the kinase domain, and blocking phosphorylation inhibits activity as Sunitharan GDP. VRK2 is actually involved with infections when working properly. It has been shown to physically interact with the Epstein-Barr viral infection proteins and vaccine protein B12 to prevent apoptosis. VRK2 is also downregulated in neurological disorders, leading to neuronal loss and increased cellular stress. A 2016 paper also found that the mutation of isoleucine to valine, which sits right next to the active site, is related to impaired prenatal development and cognitive dysfunction. Although you would assume this protein is related to cancer due to its important regulation, a 2017 review paper found that it is rarely deleted or mutated in cancer cell lines, but the expression amounts were changed depending on the cancer. Ultimately, VRK2 is involved in many signaling pathways, and it has 2 isoforms that determine its location in the cell. It helps to prevent viral infections and is regulated through post-translational modification signals. And those are my references.
It's great. Thanks so much. There's a lot of questions in the chat space. Thanks for those. I think there's a question about does ubiquitination in the kinase domain inhibit the active site or the kinase activity?
I couldn't find anything specific about the ubiquitination. It was just predicted sites.
Okay.
But it likely blocks the site from reaching anything.
Great. And is there an isoform that you think is most likely to be related to disease? Like, are the mutations found equally in the different isoforms? Do you know if there's a specific isoform that is most disease relevant?
That would be isoform 1, just because it is present in every cell, and it is widely expressed. The second isoform is usually only found in very specialized cell types, so it's not completely relevant, and it's more of, yeah, it more takes over when VRK1 isn't present in the cells.
Okay, great answers. There's more questions in the chat space for you, so please do answer those. Hannah, are you able to share your screen?
Yeah, here, 1 second.
Okay, great. We can see you and probably see your slides in a minute.
1 second. Here, I just have to approve it on my computer. Sorry.
Okay.
Oh.
I think we might move to the next speaker and let Hannah go later. Is Jiyun Lee there, please? Why don't you share your screen and we'll get back to Hannah a bit later. Okay. Great. We can see, just full screen it. Perfect. You're good to go.
Okay. Okay. Hello, everyone. Today I'm going to talk about PHKG2. It's a key player in glycogen metabolism. It encodes human phosphorylase B kinase gamma catalytic chain, and it belongs to CANK, serine threonine protein kinase family. And it has isoforms expressed in testis and liver, and it's cytosolic enzyme. When it's expressed, while it's expressed in mainly in testis,
its role in tests is not still well understood. In the liver, the kinase plays a key role in glycogen breakdown. In metabolism, the kinase phosphorylates glycogen phosphorylase B and activates it, facilitating the hormonal regulation by glucagon and epinephrine. When the kinase is phosphorylated by pKa on alpha and beta subunits, it utilizes ATP to phosphorylate glycogen phosphorylase B. This reaction is to maintain blood glucose homeostasis during fasting or exercising. Sorry. There is also potential function of the kinase in the testis. Research in rats shown that follicle-stimulating hormone activates glycogen phosphorylase, potentially providing energy spermatogenesis in Sertoli cells. Structurally, phosphorylase B kinase is a globular protein with around 400 residues. It likely lacks post-translational modification sites and disordered binding regions. The protein consists of catalytic domain, which makes up the majority of the protein, and chemidulin binding regions located at the C-terminus of the protein. It is unlikely to have transmembrane domains, even though Foveus analysis suggests a high probability of transmembrane-like regions. It is within its catalytic domain, so it's not a valid expectation. Key functional sites include ATP binding sites, probably for catalytic activity. No acetylation. Oh, okay. And here, no acetylation. Oh, sorry. Okay. For the regulation, the kinase catalytic domain occurs through its interaction with regulatory alpha subunit, and phosphorylation of the alpha subunit by pKa induces a conformational change, which in turn affects the catalytic domain. Additionally, calcium ion recomodulin binds to the gamma subunit in a compact or inactive conformation. Upon sensing calcium ions, the modulin undergoes a conformational change and facilitates detachment and inhibition of kinase domain. And no acetylation or methylation sites found, and it lacks significant lipid binding regions as well. This suggests that its regulation primarily relies on protein-protein interaction. And a few phosphorylation sites found in the protein may be linked to breast and liver cancers, but further research is needed. Mutations in the kinase are associated with glycogen storage disease type 9C. It's a condition caused by liver phosphorylase kinase deficiency, particularly in children. In affected individuals, glycogen cannot be efficiently metabolized into glucose, leading to hepatomegaly and cirrhosis. The disease follows an autosomal recessive inheritance pattern with identified variants here, and these mutations result in non-conservative amino acid substitutions in highly conservative catalytic core regions of many kinases. The altered charge of these residues may disrupt the catalytic activity of the kinase, leading to the disease. These are references. Thank you for listening.
That was great. Perfect. Any questions? Feel free to ask. I think there are a couple in the chat space showing up here. Are there some drugs that have been developed to target this kinase?
I couldn't find like a specific drug that targets this. Yeah.
Okay. And is there a domain linking it to the glycogen particles or other proteins associated with the glycogen particle?
Also, like it interacts with glycogen particle with like also with other subunits like alpha or beta subunits in the protein. Yeah, as a
Great.
like a
And
whole
are there any, are there any therapies available to treat glycogen storage disease in humans?
Saying like I couldn't find a drug or therapy associated like to treat this disease.
Okay, great. That's fine.
Yeah.
You did a great job. Thanks so much. I'll let you go and ask Joseph to share his screen. You guys are doing a great job keeping on schedule. Joseph, I see you there. Can you hear me? If not, maybe Hana, can you share your screen?
Yeah, I can try again. Sorry about that.
Okay. Why don't you go for it, and then we'll do Joseph's talk after yours.
Um,
Perfect. It's coming
Did
up.
that work? Yay. Okay.
Just full screen, and then you're good to go.
Yes. Okay. Give me 1 second. Okay. Ready?
Go for it.
Okay. Hi, everyone. My name's Hannah, and I'm doing my kinase presentation on the PX domain containing serine threonine kinase-like protein, also known as PXK. So PXK is localized to the brain and spinal cord and has shown cellular localization in the cytoplasm and has also been seen to co-localize with proteins found in both the early and late endosomes as well as the lysosomes, and it can also interact with phosphoinositides in the plasma membrane. So these are the domains of PXK. On the left-hand side is a structure from PyMol. So in blue is the PX domain, in pink is the kinase domain, and in red is the WASP2 homology domain or the WH2 domain. The PX domain has conserved basic and aromatic amino acids that allow it to bind to phosphoinositides in the plasma membrane, such as phosphatidylinositol 3-phosphate. The WH2 domain is an actin binding motif region, and it can bind to actin, allowing for cytoskeletal movement. And then the kinase domain is really interesting because it's actually completely catalytically inactive, and there are a couple reasons for this. So on the left-hand side here, these are the amino acids that are conserved in active kinases, and on the right-hand side are the mutations in PXK that make it inactive. So you have the deletion of the lysine, the aspartate to the histidine, the glycine to the glutamate, and then the absence of a glycine-rich loop and conserved glutamate, and then altogether those make it completely inactive. For functions, PXK serves to regulate the sodium-potassium ATPase in the plasma membrane, and it does this through two main mechanisms. The first one is PXK is known to be used for lysosomal trafficking and endosomal trafficking, and this helps regulate the localization of the ATPase in the membrane, and by ensuring its correct placement in the membrane, it affects its surface expression and thus controls its activity. The second mechanism that does this through is the ATPase is known to be regulated through phosphoinositide modulations. As we saw in the last slide, PXK can bind to phosphatidylinositol 3-phosphate through the PX domain, and this will alter the lipid environment around the sodium-potassium ATPase, and this will alter its function indirectly through lipid modulation. On the left-hand side is a electrostatic diagram of PXK, and this shows in blue, the basic conserved amino acids in the PX domain that I mentioned earlier. On the top right-hand side, there's a graph, and there's phosphorylation in the PX domain, and this shows that there can be altered affinity for phosphatidylinositol 3-phosphate. There's also methylation sites in the WH2 domain and acetylation sites throughout the kinase domain, and these can act as transcription blockage sites. There's also no signal peptide sequences, no transmembrane domains, no myristoylation, and no prenylation anywhere throughout PXK. This bottom graph shows the disordered regions, and it shows that there's one big one present at the end near the WH2 domain. This is important because with the disordered region, it allows for the flexibility needed for the actin binding and the movement of the cytoskeleton region. This is a graph of the motor results from PXK, and after analysis, it shows that there's membrane binding regions in the PX domain as well as the WH2 domain, and this is to be expected just based off of the functions that both of those domains serve and how they need to interact with the membrane. As for health effects, since PXK is known to be used for lysosomal trafficking and endosomal trafficking, anything that alters its effects or its ability to properly function can impair apoptosis, which leads to uncontrolled cell survival, leading to cancer and autoimmune disorders such as lupus. So that is all I have for you guys. If you have any questions, I'll be happy to answer them. So yeah, thank you.
That was great. Thanks so much. There's a question here. Does the inactive kinase site the first question is, does PXK have any isoforms?
Yeah, so PXK has 18 exons, and then through different alternative splicing mechanisms, there are 5 isoforms. The one I talked about today is PXK1, and that's the most prevalent in humans.
And then Ashwin asks, does the inactive kinase have any functional purpose?
Like as a kinase itself, it doesn't really do anything. There's no catalytic activity. It does not function as a kinase at all. I think the presence of the kinase domain helps to form the PX domain and the WH2 domains as well, because without that kinase domain in between the two, they wouldn't be able to function properly, and it just helps the whole protein fold properly.
Great. And are there any disease-related mutations in PXK?
Disease-related mutations, there aren't, and it's mostly just a loss of function. So if there's anything that alters its function, so phosphorylations in incorrect regions, or things that alter the ability to bind to phosphatidylinositol 3-phosphate that prevent it from doing the lysosomal and endosomal trafficking, will prevent it from working, which lead to cancer or autoimmune disorders like lupus.
Okay, great job answering questions, all of you, in fact. I think that's most of the questions. There might be a couple more that pop up in the chat space, so do answer those.
Okay, thank you.
Joseph, are you able to share your screen next? There, I
Yes,
can see
sorry.
you.
Earlier, I just needed to allow it to share. Sorry.
Okay, it should work now. If it doesn't, let me know. Oh, that's great. Just full screen and you're good to go.
Yep. Okay, so I'll be talking about a little bit about the NEK4 kinase, which is a
Just a minute,
serine.
Joseph.
Yep.
It looks great, but it's your presenter view. Can you put it on the full screen mode? If you go to maybe slide show, maybe it will go to, if you click on the right down tab.
Which tab? Sorry. This is the slideshow.
Yeah,
Oh, wait.
start from the beginning.
Presenter?
Yeah.
How's that?
Actually, go to start from beginning. Just below that.
1 sec.
The second option there. Yeah, perfect.
Is that good?
It's still showing the presenter view. Can you click on that? Start from the beginning tab, and then hopefully it goes. Okay, it's still the presenter view. It's okay, but if you can get it to start from the beginning maybe you try that one out. Try the presentation display options, and then full screen is what you want from the beginning. Perfect.
How's that?
Yeah, I think it'll pop up in a minute. My screen is just waiting for it to display. And you're our last presenter. Thank you for your patience, everybody. And it's still loading, so give it a couple more seconds. Probably have a lot of maybe big images or something because it's my computer isn't seeing it yet. It's still loading, still loading. While we're waiting, I want to thank you all. You've done a great job presenting. I know the time is a bit tight. We're more or less on schedule today, so we should finish before 10:50 so you can get on with your day. I look forward to the presentations on the 17th with Alessandro giving the first talk. We're going to have, I guess, one more talk that day, so we're going to try and keep it on schedule. For some reason, this isn't loading, so maybe go back to your previous view, and we'll just go with the original view, and you can just scroll through. So I'll let you start, and whenever you're ready, okay?
Yep. Sorry, so this shows the presenter view.
Well, I can see your 7 slides on the left-hand side, and maybe if you can just zoom out a little bit on your on your slide. So I don't know if you have a window kind of a way to increase your view, but it's not a big deal. I can see it, so I think why don't we just roll because it's good enough, and the main thing is just that you talk through it, okay?
Okay, sounds good. So I'll just do it this way. NK4 is a serine threonine kinase that seems to only phosphorylate threonine residues. It's expressed in most tissues, but highly expressed in the testes and followed by the ovaries It's in a cell, it's localized in the cytosol and the basal bodies of cilia, and it's involved in several cellular processes, mainly the primary cilia assembly, where it interacts with RP-CRIP1 and RP-CRIP1L in the basal bodies of cilia. Secondly, the DNA damage response and replicative senescence, where it interacts with components of the DNA-dependent protein kinase complex and facilitates them to sites of DNA damage. And this leads to a downstream activation of the p53 tumor suppressor and signaling for apoptosis and replicative senescence, which just means that it limits the amount of times that the cell is able to replicate after that point. And lastly, it's also involved in microtubule stability, but the mechanism has yet to be elucidated. This might be the reason why it's taking a little bit to load. I made an animation of the AlphaFold NE4 structure. I exported it as a GIF and it took a while, so this might be it. So it's comprised of the N-terminal kinase domain in purple, the ATP binding site I colored in orange, and also the active site aspartate residue as a proton acceptor, which you can barely see at the moment. And there's several disorder regions colored in gold. So I ran the sequence through IUPRED3 and Disolupred, and there were two areas of interest where there could have been disordered membrane interaction sites, but in the end, it didn't line up very well. So I'm going to conclude that there may not be any disordered membrane interaction sites. There's also no predicted lipidations for NEK4, no palmitoylation, no myrcillation, no prenylation by SwissPalm, Myrcelator, or the prenylation prediction suite. No signal peptides were predicted using Phobius or Filius, as shown here on the screen, and no coiled coil structures were predicted using deep coil or Marcoil. But NEK4 is regulated by phosphorylations, and I have the MODA data of the AlphaFold structure, and it suggests that there are around 26 possible membrane binding sequences, many of which are within 5 residues of known phosphorylation sites, as shown on the right here, which indicates an abundance of PIP stops. And I wanted to show where each of these phosphorylations were on the MODA data. So I've just correlated these colors here. And other than that, here are my references. Are there any questions?
That's great. Thanks so much. And there's a couple questions showing up in the chat space. Manganese, could you explain its significance? You said in the third slide that it's interacting, is that right? Actually, I'm not.
Yes.
Okay.
The manganese, it's just like the role of magnesium as well. It stabilizes the interaction with ATP.
Okay, I think that might have actually been for the previous speaker. Sorry. Is it related to any disease-linked mutations? Does NEK4 have any disease-linked mutations?
I didn't find anything directly related to NEK4 other than like cancers, which there's a few. There's colorectal. There's also lung cancers. Its interactions with RP-CRIP1 and RP-CRIP1L could lead to disorders in the cilia formation within cells.
Okay.
There's one paper on that one, yeah.
Okay, great. You've done a great job, all of you actually, on your presentations. Thank you so much. You're all very clear, and the slides were well constructed and nice and simple. Feel free to continue to hang out and answer the questions, including Joseph, if you could take a look at a couple of those that I didn't ask. And then we'll do the same thing again, I guess, on the 17th, same format. We'll begin pretty much sharp at 10 o'clock, so please do join us then. And remember that you are still graded on participation as well as, of course, attendance for these to help, support your fellow students. It's great that you come along. It's great that you all showed up today. Really appreciate it. And I'll just hang out here. If there's any questions, feel free to ask me. You can unmute and ask me anything you wish, or just email me questions as well. Really appreciate all of your effort in doing this talk today. You've done a great job. Okay, and I want to wish you all a pleasant weekend. Hopefully get some rest. Maybe do some skiing, who knows? All right, I'll just hang out here and yeah, just thanks everyone.